Okay, good morning and good evening, everyone. Welcome to join the webinar Invisible Hazards, Measuring and Detecting Airborne Pathogens. My name is Li Bing, and I'm the Head of Academic Relations at SwissNext China, Science Consulate of Switzerland in China. We connect the dots between Switzerland and China in education, research, and innovation. Today, we are very glad to launch the Connected series, a new format at SwissNext China, which focuses on the best Sino-Swiss collaboration projects and research efforts. It sheds light on how teams from both countries can work together to create synergies and get their projects off the ground. In our very first installment, we feel privileged to have two distinguished researchers in environmental engineering. Now, please allow me to introduce them. Professor Dr. Wang Jing, Associate Professor in the Institute of Environmental Engineering at ETH Zurich, leading the group of air quality and particle technology. He is also group leader at Swiss Federal Laboratories for Material Science and Technology. Professor Wang obtained his PhD degree from the Department of Aerospace Engineering, University of Minnesota in 2005. His main research interests are air quality, environmental and health impact of airborne pollutants, airborne nanoparticle measurements and emission control, air and water filtration. Now our second speaker will be Professor Dr. Gao Ming, Associate Professor of Environmental Engineering at Beijing Academy of Agriculture and Forestry Sciences. She graduated from Research Center for Eco-Environmental Science, Chinese Academy of Sciences, and has been the lead PI of 10 research projects over the last 5 years funded by National Natural Science Foundation of China, etc. Professor Wang and Professor Gao are leading the Sino-Swiss Science and Technology Corporation funded project, Emission Quantification, Transport Modeling, and Risk Evaluation of Airborne antibiotic resistance genes from key sources in Zurich and Beijing. Today, they will share with us the latest development of their studies on airborne pathogens. So at the bottom of the Zoom interface, you can find the Q&A function. So please type in your questions during the webinar and we will answer them in the Q&A session. Now, without further ado, I would like to give the word to Professor Dr. Wang. Professor Wang, the floor is yours.
Okay. So thank you very much, Li Bing. So it is my pleasure to have this opportunity to talk to a number of participants through this webinar. So I noticed among the participants, so there are former group members of mine and also current group members. So it's very nice to see you, and thank you very much for taking the time and to attend this webinar. Okay, so I'll start with the presentation. So the title I have today is Fighting the Invisible Enemy, Studies on Airborne Pathogens. So first, let me show you this, so where I took this name from, Fighting the Invisible Enemy. So after I did some search, I found that this was a book. This was a book in 1990. So actually this is a book for the young people, and to understand the effects of conditioning actually is for the psychological problems of the young people, how to deal with psychological problems. So actually at that time it was not about the viruses or pathogens, but of course today also a lot of the news media are using this phrase for the current pandemics of the viruses. So I just want to make clear that I'm not quoting Donald Trump in this case. Okay, so so here's the outline of my talk. So I will talk about the pathogens and the air quality, specifically on the antibiotic resistance genes and endotoxin and the global distribution of these pollutants. Then I'll talk about biosensors for these pathogens, more generally for the total bioaerosol and for the recent SARS-CoV-2 detection. So after we do these measurements, and then we also would like to mention a little bit on the antibacterial technologies, how to kill the bacteria. Okay, so first, so the air quality, I believe everyone knows very well. In China, especially, the air quality is heavily dependent on the particulate matter. So the PM 2.5 or PM 10, this is basically air quality indicator that everyone is paying attention to day after day. But generally speaking, the government regulations are for the total mass of the airborne pollutants. So our intention in the recent years is to highlight that the health impact depends on the components of the particulate matter. It's not only the total mass concentration, but also what constitutes the particulate matter. For example, we know that heavy metals, these are quite toxic. And if you have endotoxins, we'll talk about that, and the bacteria, the viruses, and now we know very well, even only a tiny amount of these airborne pathogens could cause significant health impacts. So in addition, you could also have other ones, organics, fungi, and the inorganics. If you only have like ammonium salts and things like that, maybe it's not so bad. But if you have a lot of these pathogens, then you need to really worry about the air quality. So in this talk, of course, we have the focus on the pathogens. So I will mention a little bit about the background of bioaerosols. So bioaerosols means the airborne particles with biological origin. So it could be from the human, could be from the nature, including environment, the animals, the plants. So these could be from plants, soil, water, and animals. They could also serve as sources for bioaerosols. The categories of bioaerosols include bacteria, mold, virus, pollen. So they have different sizes and different characteristics. If only a tiny amount of these pathogens, when they get into the human, they could cause significant diseases, infectious diseases, respiratory diseases, cancer, so on and so forth. So two of these, two of these pollutants. One is the endotoxin. Endotoxin is also called lipopolysaccharides. So these are big molecules, which is part of the membrane of gram-negative bacteria. So these, so even if the bacteria die, their membranes could survive, and if they go to the human lung, it could cause pulmonary diseases, asthma-like symptoms, and chronic bronchitis. Another one actually is the focus of our Sino-Swiss project, which is the antibiotic resistance genes. So we know that humans have used a lot of antibiotics. So these antibiotics are used to treat the diseases for humans, and humans use those, and humans also use this for the husbandry, the animal husbandry. For example, in China, I know some of our colleagues in China, they do not eat chicken because they say, okay, if you eat 1 kilogram of chicken, you eat half of the antibiotics, so that's not very good. So if you use too much of antibiotics, then the unused, the extra antibiotics get into the environment. For example, some people flush this into the toilet, and these go to the wastewater, and then in the environment, the bacteria are exposed to the antibiotics, and then these bacteria could develop the antibiotic resistance genes, and then you have, in the future, you have super bugs, things like that, which are resistant to the antibiotics, and then they could have cause the diseases which is difficult to treat. So we performed studies such as global survey of ARGs and endotoxin in the airborne form. So for example, we collected the airborne particles from 19 different cities around the world So these words including, for example, Chinese cities, Beijing, Shanghai, and the American ones, the European ones, the Zurich. And so here, first, if we look at the airborne concentration of PM10, PM2.5, then Beijing and Shanghai could be quite high, over 100, sometimes over 100 PM10, sometimes PM2.5 is also very high compared to the San Francisco, for example. This is much higher compared to the European countries. But however, if we look at the antibiotic resistance genes, the distribution is not always the same as the PM. For example, in San Francisco, from this heat map, you can see the concentrations, these red ones, both these individual ones, against beta-lactam, against the tetracycline, against quinoline. So these ones are relatively high. Also, the total concentration is relatively high. Beijing is also relatively high on the list of antibiotic resistance genes. And then down here, a relatively clean environment we see in Zurich. Zurich is relatively clean, not so much of antibiotics or antibiotic resistance genes. So basically, we look at this correlation. So this correlation on the x-axis, you see the global antibiotic resistance, global antibiotic usage. So if you use more of these drugs, if you use more antibiotics, so this is correlated with the developed abundance of the airborne antibiotic resistance genes. So if you use more, then the more of the resistance genes will be developed by the bacteria. So this is the number of detected ARG, the subtypes. So according to the subtypes, the Beijing is also quite high, so indicating that a lot of different types of drugs are being used. And Zurich is relatively low, near the bottom of the range. The beta-lactam resistance genes was found to be most abundant, seconded by the quinoline resistance genes. So the next one is the endotoxin. We measure endotoxin, the total suspended particulates. So from this one, we do not see a very clear trend. We cannot say that, okay, in Beijing is significantly higher in other locations. But this one is the unit mass. The unit mass, we do not see a significant difference. But if you multiply this with the airborne concentration of PM2.5 or PM10, because the airborne concentration is much higher in the Asian countries, after you multiply this with the unit value, then you get, okay, the Asian countries are still showing the higher endotoxin concentration in the air compared to the European countries or the North American countries. So not only the endotoxin, we also measure the toxicity. So here the toxicity is indicated by the oxidative potential. As we know, the oxidative stress is one of the most important mechanisms for the particles to induce the toxicity in human cells. So the toxicity, again, we see that the Beijing toxicity of these particles is not really higher. Actually, the San Francisco particles showing higher toxicity than Beijing. So again, now you multiply the PM2.5, PM10 total concentration in the air, the Asian countries are showing higher toxicity than the European and the American cities. We continue to the next topic, and then we try to develop sensors to do faster detection and measurement of these pathogens. So different sensors are already available methods, for example, the culture-based methods. So these one can be used for viable bacteria. So the viable bacteria can grow, and then you can use your naked eye identify these things. Or you can use the PCR. So PCR is polymerase chain reaction. So as you know now very well, so this is basically the gold standard for diagnosis of COVID-19. So if you have the virus of the coronavirus, then this is basically a standard method. You're going to use this one to identify the RNA of the virus. You could also use fluorescence-based method to do real-time detection of bioaerosol, atomic emission or mass spectrometry. So these can measure the elemental composition, and they also have some limitations because the interference of background and it does not give you specific information of the bioaerosol. So the plasmonic method, I'll give a little bit more detail on the plasmonic method. So the plasmonic is a phenomena quite often related to the noble metal nanostructures. So for example, in this one here, we have gold nanofilm, and on the gold nanofilm, we have gold nanoparticles. And if you have an incoming light, the electromagnetic field of the light will have interaction with nanoparticle, then you have an enhancement of the local electromagnetic field, and then this one can provide you a signal, and with this signal, you can do the sensing of the certain targets that you are interested in. So this is the plasmonic system in our lab. So here is the sensing element. With the sensing element, you can see the magnified image, you can see the gold nanofilm, and from this AFM scanning image, you see the gold nanoparticles. So with the gold nanoparticles, so our team have performed the optimization to optimize the nanoparticle size and the gap of these nanoparticles, eventually try to enhance the sensing signal. And then the other ones are basically standard optical instruments, try to use the light to the sensing part and then get the signal. So the first example I show is for the total bioaerosol detection. So for the total bioaerosol detection, we are targeting the amino group on the biomaterials. So to capture the amino group, we do the functionalization on the gold nanoparticles with the theo group. So the theo group will have the functional part and then modified with EDC and AH, and then this functional group is ready to have the covalent bonding with the amino group, and then by this we can capture the biological material. So this is the calibration. The calibration, we can use E. coli to do the calibration. We can also use the Bacillus subtilis. And with this calibration, we obtain the linear range. So we can get the signal. And then with this sensor, we perform the practical measurement at different environmental monitoring stations of Switzerland. So these stations, including this suburban station in Basel, this urban station near roadside in Bern, and also this specific station on the Rigi mountain. So on the Rigi mountain, this is above 1,000 meters, so at high altitude and quite clean background. So the measurements from this heat map, you can see basically here is showing our measurement result for different months. So for different months, the general characteristics you can see. So in these stations in the winter, you see quite low concentration. This concentration is getting higher in the summer because in the summer, the biological activities increase. So we have higher bioaerosol concentrations in the summer. And in the mountain Rigi, this concentration is quite low for the whole year. So now if we plot these the concentration, so this concentration, the signal from our biosensor plotted together with the endotoxin concentration, we see a very good correlation. So because both of these biosensor signal and the endotoxin signals are due to the biological activities, so they have very good correlation. But if we look at the concentration of PM10, so we know that the air quality is generally not so good in the winter. So the concentration is high in the winter and low in the summer. So the correlation is anti-correlated. So now we come to the virus part. This is the coronavirus. So we know the coronavirus, you have the outside envelope, there are proteins here, and there's RNA inside. So the standard method is the PCR method. PCR, basically, you control the temperature, and then you amplify this, and then you finally get a signal. Then you know whether you have this virus or not in your body. So what we want to do is have a plasmonic sensor. So this plasmonic sensor is not only, previously I talked about the electromagnetic field, but also with the photothermal effect for the temperature control. So now on the top of this system, we added one with this laser. You can use this laser to control the temperature. As we know, the DNA and RNA hybridization is important, dependent on the temperature. So this is the target RNA of the SARS-CoV-2 virus. So to show that we can have a very, very accurate selective detection of this virus, we try to compare this with a very closely related virus, which is the SARS-1. So we selected different parts of this genomic sequence, and then with this genomic sequence, so this is our target genomic sequence of the COVID-19 virus. This is our functionalization with our DNA. So if they are perfectly matched together, then by controlling the temperature, when we increase this temperature, then we have a much faster response and get much more accurate detection and also much more sensitive detection with this specific virus. However, this is with the genomic sequence with the SARS virus, the SARS-1. So this is not our target. There are three different bases, which is not a perfect match to our DNA. So this one, when we increase this temperature, actually this not perfectly matched virus sequence will be de-associated. So initially at the room temperature, actually you can have some temperature, you can have some hybridization with our DNA, but at higher temperature, we can actually remove these things from our sensor. So with this, so we can do the concentration gradient and find our lower detection limit. So our detection limit of the sensor is about 10,000 copies. And then this is enough for the diagnosis because the typical range of the throat swab samples is from 10 to the 4th to 10 to the 7th copy. So this is good enough for diagnosis, but our goal is not only to do the diagnosis, but also to use this for environmental detection. So for example, we know that these viruses could be floating in the air for transmission of the virus. We would like to do that. And then whether this lower detection is enough for the environmental samples or not, we still need to do more research to find out. So this is a video made by the Swiss radio. So this is in our lab, and so our people are doing the detection of this virus. As you can see, the laser, the sensor, and you can get a little bit sense of how it works. So this is Guangyu, this is Rang to do this detection. Well, basically you can see here a little bit, get a little bit sense of how it works. Okay, so final title is on the antibacteria. So I have very limited time, so I will just very quickly go through with some of these so for example, we can use the different filter materials, charged filter, or the chitosan to kill the bacteria. So the bacteria will lose the activity. And we also have metal-coated filter samples. For example, we know copper is efficient to kill the bacteria. So we can put a copper layer, and then this is a membrane, a nanofiber membrane. And then with this nanofiber membrane, so with this one, with the fluorescence signal here, so fluorescence, the green ones are the live bacteria, and then after we put these bacteria on the copper nanofiber, then they are killed and basically showing this red color. But I don't have the time to go through all the details. So in the end, I would just like to thank all my group members. Obviously, all the work, all the research was performed by my group members. So So I would like to take my group members to enjoy the nice air. So we go up to the Swiss mountains. We still have some nice Swiss mountains to explore. So thank you very much for your attention, and thanks for the host.
Okay, thank you, Professor Wang, for the interesting talk. Now I'm delighted to invite Professor Dr. Gao Ming to give her talk. Professor Gao, the floor is yours.
So, hello everyone, and thank you very much for attendance, and thank you for leading. And I'm very honored to be here. My name is Ming Gao. I'm an associate professor from Beijing Academy of Agriculture and Forest Scientist, and I feel so honored to be here to introduce you guys some latest result on the characteristic of the animal-related antibiotic-resistant bacterial also. So first of all, I want to introduce some background of antibiotic resistance. And in 1900s, when the discovery of the antibiotic happened, it solved tons of problems result from the bacterial infection. So, but however, because this new antibiotic can give the selection pressure for the pre-existing antibiotic resist genes, that genes maybe exist for about 1,000 years. This resulting the suddenly development of the globally antibiotic resistance happen. So according to the report from the WHO, currently at least 700,000 people die each year due to the drug-resistant disease. If we did not do anything, in the next 30 years, this number will reach to 10 million per year. So now we want to know how the antibiotics resist happen and why it seems unstoppable. I did a simple fingers to show you that what's this process happened. Here we use we called antibiotic resist is called for short AR, and antibiotic resistance is called for short ARG, and antibiotic resist bacteria for ARB. These two nucleic acid, the red and the blue one is present the active ARG in the cell, and this green one is present the inactive ARG. Under the selective pressure of the antibiotics, the Asian inactive ARG become activity. Now some people will ask that if we stop use antibiotic, this ARG will disappear. Actually, it's hard to say because some ARG will persist for a long time because of the horizontal gene transfer. We call it HGT. So what HGT is? It's really easier for us to understand that genetic characteristic usually be transferred from our parents to kids. But with the help of the HGT, the bacteria can transfer their genetic character to their friends. And their many friends can use the same way to transfer this ARG to their friend and their kids. This process happens again and again. That's why the process, it seems developed so quickly and it seems unstoppable. So we usually care about what happened about the AR in the clinical settings, but there are some places that we need to pay attention. according to the report from the Science published last year, that 73% of antibiotics sold on Earth are used in animal farms. It provides an ideal selection pressure for the enrichment of ARG and ARB. So, under so much select pressure, what happened to the bacteria related to animal? So, according to the Lancet Infection Disease, they published a paper that their result indicated that MCR1, the gene against clostridium, a drug of last resort, was first reported at Chinese swine farm. During the last 20 years, the proportion of ARB continuously increased in chicken, pig, and cattle farm. So after knowing all of this information, we realized that ARG in the animal farm is a serious issue. So now let's talk about another keywords of my presentation, the biosol. We can call biosol is kind of biological stuff in the air. It's simply said. I mean, that is that thing. I mean, so maybe you don't know that the Professor Wang introduced. There are lots of ARG was detected in whole world globally. I mean, so maybe you don't know that the ARG in urban air, their relative abundance is comparable to the wastewater treatment effluent, and is higher than drinking water. Some results published from EST last year indicate that in some cases, the ARG contribute to the similar human exposure as the drinking water. After I told you that ARG in the air of the farm, I mean, so their concentration is far higher than the ARG in the air of animal farm. Maybe you will realize that around the animal farm, that you are expected to be exposed far more ARG than in urban. Some people believe that if I avoid the animal farm, so the risk of exposure for the ARG will be decreased. But it's not quite true, and it's not the whole story, because ARG can attach on the particle matter, and they can be transported by wind for long distance. Not only the biosol from Asia country can be transported to the West United States, and but also the entire microbe community from the Sahara can be transported to the European. Using modeling method, it means our latest result from the cooperation with Professor Wang Jing indicated that chicken farm had certain contribution to the ARG in the most urban air. So based on all of this information, this can help us to understand that even you live far away from the animal farm, you still can suffer the ARG, I mean, transfer from there. So next, I want to talk about what do we describe the status of the ARG and ARB related to animals. As we understand that the airborne ARG in the farm have inevitable risk. So we want to know what it consists of that and where it is going, what effects it may have on Earth and on the environmental. So to solve all of the problem, it requires in-depth acknowledgement of the characteristic of animal-related antibiotic-resistant bacteria also. So that is the work that we have been to last 5 years. So this is today's story and a part of our story. I want to introduce part of our work to you today from two aspects, genotype and phenotype. The genotype is the individual traits. It's and the genetic contribution of the phenotype is a genotype is called the phenotype. So it's a relatively complicated, but I can explain it to you from this way that the ARG is kind of genotype, and the AR is, and that means the antibiotic resist ability is related to the phenotype. So we just want to build to the connection between the genotype and the phenotypes. At the first beginning, and we are do tons of work to try to get enough information to figure out how many and what's the concentration of the ARG in the air of the animal farm. And then we want to find out how this ARG to influence some pathogens. In our study, we take the Staphylococcus aureus as an example to study. So if we wanted to get all of this information, we needed to do the first three part of the work. Yeah, that says first you need to find some place to get sample. It's most dependable farm, and second, you need to collect enough sample, and the third one is important, that you must analyze the sample very well. So let's begin the first part of our story, and then the genotype. So at the very beginning, we do research on about diversity of two types of ARB in Bialso and manure in 4 types of animal farm. We almost collect about 200 samples from 18 animal farms. Among all of the interesting results, I want to share with you one that about the Staphylococcus. It is a key genus in antibiotic resistant bacteria, and their relative abundance in the air is significantly higher than in manure. So it seems that that strain is very important in air compared to the manure. Now we do some research on the qualitative analysis of ARG. This part we use the digital job PCR to detect the absolute concentration of the 45 subtypes. This time we collect about 75 sample, and to detect the concentration of the ARG from the manure indoor and outdoor. Based on our results, we first found that we detect the correlation on ARG between the animal farm and the urban. So if we want to get enough information, almost the whole information of the ARG related to animal farm, we need to use others methods, something like metagenomic analysis. So use this way that we analyze the almost 100 samples to totally we got about 2,000 ARG types information that were resistant to 15 major classes of antibiotics. I know all people care, they're curious about what's the situation in the urban air. It's about 800 type of ARG. After we compare the difference between the animal and the urban, we found that in the city air, there are only 22 unique ARG was detected. So this part of result, we can suppose that maybe animal farm is the one of the important source for the ARG in the urban air. So Let's begin the second part. We want to talk about phenotype. We want to analyze the resistance, their influence on the evolution of this pathogen. So according to our previous result, this time we collect the sample from different provinces in China based on the published antibiotic antibiotic distribution actually. We isolate about 500 strains. Here I just give you some information on our first 300 strain. We detect their antibiotic successful test to 17 antibiotics. The result is very interesting that all strains will resist to penicillin. That means that the penicillin doesn't work to kill this strain. And the good news that that none of them resist to the vancomycin. Yeah, vancomycin. This is kind of good news that we all know that vancomycin is the kind of last drug restored for gram-positive bacteria. So we also detect the multiple antibiotic resist ability using the index MAR. We found that the index of this strain increased with the time increase during the whole 1.5 years experience. Our primary result indicates that farm environment could affect the evolution of the pathogen. So we also do some work on the genotype of this bacteria. This is the according to the result, we got the similar trend. Here we shared some very interesting variation of the genotype and the phenotype. I want to say that although our group spent a lot of time on this research field, it's still a very initial result. I expected to find a more interesting result after we finish all whole gene sequence analysis. So the last part is what are we going to next? Here we know more about the characteristic of the animal-related antibiotic resistant bacteria also. So we can see that some of the pathogen in the air before is invisible, now it's kind of visible. And now we can understand some of the questions I asked at the previous day of the presentation. And after we got this data, we will do a cooperation with Professor Wang Jing, and we want to evaluate the ARG emission from the animal farm and to quantify its contribution to urban air. And based on this information, and we want to do some work with the Chinese CDC and to evaluate their health risks. Yeah. So I want to thank SwissNext to give me this opportunity to share some results with you. And I want to see Vivian and Tobias do tons of work for this. And just say thanks to this funding to support my research. And thank you for your time. And any question? Yeah, thank you.
Yeah, thank you very much, Professor Gao, for the informative talk. Now let's come to the Q&A session. I see that we have received a lot of interesting questions from the audience. So if I may ask you to try to keep your answer short so that we can cover as much of them as possible. So first question from Bing Xu to Dr. Wang, Professor Dr. Wang. Could you share more about the mechanism of the biosensor related to the nano gold layer? And he also asked, I saw a radical group grafted on the AU molecular. What is the reason to choose that one, please? Thanks.
For the sensor, basically the sensor, the gold nanoparticles is the platform to generate the plasmonic effects. So we have this plasmonic effect, and then the gold nanoparticle need to be functionalized according to the specific target you would like to detect. So for the specific target, then you choose what type of functional group you graph onto the gold nanoparticles. For example, for the virus, this is the cDNA, which is corresponding to the RNA you would like try to capture. So this hybridization is very specific. So then the cDNA is specifically corresponding to the target RNA, so they can have this hybridization. Once they have this hybridization, then the local refractive index is changed, so the plasmonic effect will change. Then we have a signal, and this signal gives you the information if this target is detected and how much is being detected.
Okay, thank you. Then one question for Professor Gao from Lu Fan. Thank you for interesting works. Might I know the amount of the bioaerosol or air sample to be collected for metagenome analysis? Thank you.
I means it's depends. Different sample. I mean, it's different. At most, it's about 5 ng. It's 5 nanogram, I think. It's, yeah.
Okay, thanks. Then next question, when can the cutting edge sensor technology be applied into practice, and how long does it take? This would be for Professor Dr. Wang.
Well, so right now, as you saw in the movie, this instrument is still a lab instrument. So right now, you still need to manually feed the samples into the instrument, and the signal is manually collected and analyzed. So we still need a number of steps to have the sampling, and then to directly input the samples into the sensing cell, and also to collect the signal to do automated signal analysis. So all these procedures will need some more time, and we are still trying very actively to acquire more funding to do the development. And so this will take some time. But of course, the development will be dependent on how much of the manpower, how much of the resources we will have for this develop. So I will say that we can definitely have a unit that we can deploy in the field let's say in the next within 10 months or something like that. When this will become a commercial instrument, this probably will take longer.
Okay. And a follow-up question, do you have planned to do research in the Swiss hospitals on the disinfection of air and the surface? This is from Michelle Ma.
Right, so right now we are, well, I have been in contact with I don't know, maybe 20 doctors, maybe not that many, maybe 10 doctors from different hospitals, from University Hospital in Zurich, from Geneva Hospital, Lucerne, Canton Hospital, Winterthur, a number of these doctors. So we have been discussing the possible collaboration, and we also have proposals with the doctors doing these tests in the hospital. So we have plans to do this, and so far we have not done that yet. So far we're still trying to improve the sensor so that the sensor can really be a standalone unit which can be deployed in the field.
Okay, thank you. Then next question for Professor Dr. Gao. What is the direct source of the bioaerosol from farm animals? Is that coffee?
Yeah, we suppose this come from the manure of the animal, because after the animal get rid of their manure, the manure just stay in the animal house. It's just stuck at there. So this bacteria in the manure will be aerosolized and emissioned in the air of the house. So that's why we detect that things. It's I think a cough will contribute part of them, but not the most of them. Yeah.
Okay, thank you. Then another question for Professor Dr. Wang. I found that the sensing mechanism towards virus SARS-CoV-2 and E. coli, if I pronounce it correctly, are quite different. Can your sensors detect SARS-CoV-2 directly rather than the extracted RNA, like what you did for the detection of E. coli? Thanks.
Right. So yeah, certainly developing different types of sensors. So for the sensor, you can try to target the RNA. You can try to target the virus as the entire entity, and then using antibodies which directly conjugate with the virus. So this depending on what is your preference, and then you can certainly craft different types of antibodies to achieve the direct detection of the virus.
Okay, thank you. Then the next question for Professor Gao. What were some of the biggest challenges you encountered when trying to measure animal-related pathogens, and how did you overcome them?
Yeah, I think we all know that the concentration in the air of the pathogen is very low. So we just try our best to collect enough sample, as I mentioned in the presentation. And sometimes, because we have enough time, it's different from the situation about Professor Wang Jing's research. They can detect this very quickly. But we use a long time and to analyze it, use some machine like a digital job, the PCR, and to detect a very long concentration of that. So that's it. The first day you try to concentrations a lot of sample, and then use the high sensitive time of the machine to analyze it. Yeah, that's it.
Okay, thank you. Then some follow-up questions from Bing Xu to Professor Wang. What are the criteria to select the grafting functional group on the AU atom, please? Polarity of the radical group or some other properties of the grafted functional group? Thanks.
It's dependent on the specific target you would like to capture. So, for example, we use, as you saw, when we try to develop these sensors for the total bioaerosol, this is one type of functional groups. And when we try to detect the virus, this is a different types of groups. So it dependent on the specific target. And then usually we try to have you need to consider different aspects. You would like to have a specific detector so that then the selectivity is very good. So this is very specific. For example, as I mentioned, then this DNA and then RNA hybridization is highly specific. We can differentiate SARS-CoV-1 and from the original SARS. You could also have some more general functionalization. For example, for the total bio, then we are targeting the amino groups, the ubiquitous of the different types of biological materials. So that you could consider both of these. Some of these would improve the sensitivity. Some of these would improve your selectivity. Specifically, if you have a very good reaction between your target and your functional group, then you will be able to achieve a better result.
Okay. Then one question for Professor Gao. How many drugs do we still have which would be effective against the most immune bacteria you detected? and how worried are you about your findings?
Yeah, I think as I mentioned in the report that the situation is different for different bacteria. For example, the clonstein is work very well for the gram-negative bacteria. It still work. And for the gram-positive bacteria, so we usually the one cool message is the doctor always pick up this medicine. So until now, although we found very normal antibiotic resist for different pathogens, but we still have some antibiotic to use. And super bug happens, but not always happen. So yeah, people don't need to worry too much about this. And the good news that if we stop use antibiotics, the antibiotic resistance will decrease. And also the academician Shen Jianzhong, he did excellent work at this this aspect. Yeah, he found that after stop use the antibiotic, the antibiotic resistance significantly decreased. So it's the good news. So yeah.
Thank you very much. Then maybe one last question to Professor Dr. Wang from Lu Fan. So when you did the bioaerosol experiment relating to virus, do you need to operate in a biosafety cabinet, or it can be done as normal environmental samples? A clean room is enough? Thank you.
In our lab, we have biosafety level 2 lab. So these were performed in a biosafety level 2 lab. So biosafety lab, not exactly the same as a clean room. So there are different considerations. I certainly will recommend that you consider these biosafety labs if you would like to perform experiments related to bioaerosols.
Okay, thank you. So due to time constraints, we will have to end the Q&A session. Now I would like to thank Professor Wang and Professor Gao for your available time and your deep insights. Thank you very much for joining us in our first installment of Connected series. Then I would like to kindly invite you to say goodbye to our audience.
Thank you.
Thank you.
Thank you, Libin. Thank you.
Thank you.
Thank you for all the participants.
Thank you. So in the very end, you will see a slide here. So if you have more questions, please send them to the email address on this slide. We will transmit the questions to the professors and give their feedback to you. You are also more than welcome to scan the QR code and follow us on LinkedIn, WeChat, or subscribe to our newsletter. Thank you very much for your active participation, and looking forward to meeting you in our next events. Thank you. Bye.